Hello everyone, and thank you so much for joining me tonight. My name is Amanda, and I'm a PhD researcher at the University of Aberdeen, based in the Institute of Medical Sciences. My main area of interest is developmental biology, which is the science of how embryos grow, so how humans are made. Today, we're going to see what we need to make a human. So what is a human? Humans are described as bipedal primate mammals. All this means is we are apes that walk on two legs. But there are also other primates that walk on two legs, so what makes us different to them? I guess it depends on who you ask. Some people say we are human because of our intelligence. It could be our curiosity, our need to explore the unknown, or maybe it's our ability to problem solve. But if you ask a medical researcher, they will tell you the reason we are different is because of our genes. But what is a gene? Every living thing, including you, me, plants, dogs, cats, and so on, is made up of incredibly small building blocks called cells. Cells make up almost everything in the body, such as the skin, the heart, the eyes, the bladder, the stomach, and so on. However, we can't see these cells with our own eyes because they are so incredibly small. In fact, there are more than 37 trillion cells in the body. Inside of the cell is something called a nucleus, and inside the nucleus is DNA. DNA stands for deoxyribonucleic acid. Every single person, including you and me, have their own DNA, which is unique to them. Your DNA is different to your mum's, your cousins, your friends, and me. The DNA looks like this, and it contains segments called genes. Genes tell your body what to make. So, for example, everybody has a gene which tells the body to make skin, a gene to tell your body to make heart, a gene to tell your body to make eyeballs, for example. Even though your DNA is unique to you, your DNA will be 99.9% the same as every other human's. It is the 0.1% that makes us different. There was a project in the 2000s called the Human Genome Project, which was an international effort to try and understand all of the human genes. This project discovered that humans have around 20,500 genes. So that answers the first part of our question. To make a human, we need genes, and we need about 21,000 of them. But we can't just throw genes into a pile and it will make a human. So what else do we need? To make a human, we need to get genes from a man and from a woman. As I said before, genes are found in DNA. DNA is found in the nucleus, and the nucleus is found inside a cell. So we can combine a cell from a man and a cell from a woman to get the genes that we need. The male cell is called a sperm cell, and a woman's is called an egg cell. When the sperm and the egg combine, they make a fertilized egg, and the genes from the two cells get mixed together. The human, who then grows from the fertilized egg, will then have a mix of genes from their mother and their father. Hundreds of years ago, in the late 1600s to the 1700s, scientists thought that a miniature human was found inside the sperm. This theory, known as the preformation theory, stated that a tiny human with arms, legs, and all of its organs would be inside the sperm and would be activated once it found its way inside the mother. Only then would it start to grow larger and make a baby. We now know that this isn't true. Instead, the combination of the sperm and the egg cell makes new cells. But what does this look like? This is what a cell looks like. Right now, there is only one cell. This cell can split in the middle and divide to make two brand new cells. These two cells can split again to make 4 new cells. The cells keep on dividing until we have enough for the next step to make a human. It isn't always easy to combine a sperm and an egg, as the man or woman may have some fertility problems. However, a technique called in vitro fertilization, or IVF, is available to help these people. For IVF, an egg is surgically removed from the woman and fertilized with sperm in a laboratory. After 5 or 7 days, the fertilized egg is then returned to the woman's uterus to continue developing into a baby. There are 6 main steps involved in IVF. The first is cycle suppression. The woman is given medicine to regulate her menstrual cycle and to stop her from having periods. The second step is an egg supply boost. The woman is given more medicine so that she can produce more eggs. Normally, a woman only produces 1 egg every 4 weeks. The third step is egg maturation. Even more medicine is given to prepare the egg for fertilization. The fourth step is egg collection. A needle is used to remove the eggs. Then the fifth step is egg fertilization. There are 2 main ways that the eggs can be fertilized. Conventional IVF involves mixing the collected eggs with sperm in a petri dish. The fertilized eggs, called embryos, are grown in the laboratory for up to 5 or 7 days, and the best 1 or 2 embryos will be selected. Sometimes the sperm cells aren't as healthy as they could be and struggle to fertilize the egg, so instead, 1 sperm can be injected into the egg. This is called intracytoplasmic sperm injection, or ICSI. Around half of the couples who undergo IVF use ICSI. The final step is embryo transfer. 1 or 2 fertilized embryos are placed back in the uterus. It is also possible to freeze unused embryos in case the first round of IVF did not work. IVF is not just for couples who are struggling with fertility issues. It is also possible to undergo IVF if a single woman wants to be a mother and uses donor sperm. It is also available for same-sex couples. To recap, when we're making a human, we first need genes. Half of these genes come from a father's sperm and a mother's egg. We also need cells. These cells come from the fertilized egg, which can then divide to make more cells. For humans to survive, we also need oxygen and nutrients. So how can we give this to a human that's growing? Inside of the mother is an organ called the uterus, which is also known as the womb. As the fertilized egg is dividing to create more cells, it implants itself into the side of the uterus. At this point, it is called a blastocyst. When the blastocyst implants, the part that is touching the uterus wall will go on to form the placenta. The placenta acts as a bridge between the blastocyst and the mother, allowing the mother to provide oxygen and nutrients to the cells. This is what the blastocyst looks like. The trophoblast will go on to make part of the placenta. The other 2 parts, called the epiblast and the hypoblast, will go on to form the embryo itself. At this point, the embryo is bilaminar. This means it has 2 layers. To make a human, these 2 layers need to make 3 layers. It does this by a process called gastrulation. The cells in the epiblast layer start to migrate down between the epiblast and the hypoblast and form a middle layer. The embryo is now trilaminar. This means it has 3 layers. These 3 layers are called germ layers, and the different germ layers will go on to make different parts of the body. The top layer, called the ectoderm, will form the nervous system and skin. The middle layer, called the mesoderm, will form the muscles, connective tissues, bone, heart, kidneys, and reproductive tissues. And the bottom layer, called the endoderm, will form most of the digestive and respiratory system. So now we know we need the following to make a human: genes, cells, oxygen and nutrients, which can be transported through the placenta, and germ layers to make all the parts of the human body. As a scientist, I'm really interested in watching the human grow, but it's usually quite difficult. The human is growing inside the mother's uterus, so we can't see it. and most mothers don't really fancy having some scientists looking around their area. Instead, scientists use animals to learn about how humans grow. Even though earlier we learnt that animals are different to humans because of their genes, they are still useful. So, let's play a game. Here we've got 5 pictures. One of them is human and the other 4 are animals. These embryos all look very similar, so can you guess which one's the human? It's difficult to know which is which because so many animals and humans develop in the same way. This one was the human. The other embryos are a chicken, fish, a bat, and a cat. They all look very similar, and this is why studying animal embryos in a lab is useful to learn about humans' development. As we saw, embryos may look similar during development. However, this only applies to the earlier stages. As the embryo develops further, there are more obvious differences between the different animals. We can study the similarities and differences between these embryos to learn more about evolution. Embryos which look more similar during longer periods of their development are said to share a common ancestor. For example, both a fish and a salamander have gills during the mid-stage of development. This means they both evolved from an animal which also had gills. Evolutionary scientists regularly use embryos as evidence to support the theory of evolution. Let's have a look at a chicken embryo. Chicken embryos grow inside of the egg and hatch after 20 to 21 days. Chickens were one of the first embryos studied because it's so easy to look at them. I'll just open an egg and show you how easy it is to look at a chicken embryo. When I'm finished looking at the chicken embryo, I just seal up the egg using sellotape. The chicken embryo can continue developing as normal. Here is a young embryo. Can you see its heart beating? This one's a little bit older. Can you still see the heart? One area of science that chicken embryos are incredibly useful for is studying how limbs grow. By limbs, I mean arms and legs in a human or wings and legs in a chicken. The limb starts off as a bud at the side of the embryo. This bud starts to grow out and bone cells start to form. The bone cells condense together and form the bones in the limb, which are the humerus, radius, and ulna. The fingers, also known as digits, are also formed. By studying chicken limbs, we learn a lot about human limbs. A lot of the genes are the same, they grow in a very similar way, so that's why chickens are very useful in this area. We can use a range of different animals to learn about human development. For example, embryos of a fish called the zebrafish are see-through. This means we can have a look inside them whilst they're growing. We can see their organs growing, we can see their heart, we can see their blood vessels. There's a special type of fish I work with, and these are fluorescent. But what does fluorescent mean? We first start off with a special light bulb which shines blue light onto the fish. The fish then shines green and actually starts to glow in the dark. This is fluorescent light. Let's have a look at one of the fish under the microscope. So here's one of the fish embryos. If I zoom in a little bit, we'll be able to see the heart beating. There, can you see the heart? Now if we turn on the special light bulb, it shines green. Look at the heart now. So here's the fish before it's fluorescent, and here it is afterwards. So why do we care? Why do we study how humans grow and what we need to make humans? Because sometimes things can go wrong. For example, a limb might not form properly and you might have more than 5 fingers. Sometimes a baby can be born with organs outside of its body. By studying how a human is made, we can learn about all the things that go wrong. And by learning about the things that go wrong, we can figure out ways to fix it. So this now leads on to the work that I'm interested in. When a woman is pregnant, sometimes she can suffer from morning sickness. In the 1950s and 60s, if the morning sickness was particularly bad, the woman could be prescribed a drug called thalidomide. At the time, doctors thought that drugs and medicines could not reach the baby, as the placenta would act as a kind of shield and protect the baby. But it turns out thalidomide could reach it, and it was very capable of causing damage. The most common type of damage were limb defects. The arms and legs could be shortened, twisted, they could be missing bones, or they could just not grow at all. Here is an X-ray of a child's arm who was exposed to thalidomide when they were still growing. You can see they've only got 3 fingers, and they're missing some bones, such as the ulna and the humerus. We can use chicken embryos to learn about how this happens. For example, we can add a drug called CPS49, which is very similar to thalidomide. This causes the chicken to not grow the limb. You can see that the untreated chicken is normal. By studying what genes are missing or what genes are not normal, or even why the bones and the veins and the arteries haven't grown, this will help us to learn why thalidomide did this to humans. Today, thalidomide is no longer used to treat morning sickness, but it has other properties which are useful. Thalidomide is anti-angiogenic. This means it stops blood vessels from growing. This could be useful when trying to treat cancer. Some cancers need to grow blood vessels to receive nutrients and oxygen, so by preventing this, the cancer can be stopped. Thalidomide is also anti-inflammatory. This means it reduces inflammation in the body. Inflammation can be seen in conditions such as inflammatory bowel disease, transplant rejection, and some types of arthritis. Thalidomide might be a useful treatment for these conditions. Thalidomide is also immunomodulatory. This means it can be used to control the immune response. Examples of when thalidomide can be used include multiple myeloma, Crohn's disease, and complications of leprosy. By studying thalidomide and making different versions of it, we can try and design a drug which can be used to treat these conditions. So, how do you make a human? We know we need genes, which comes from the father's sperm and the mother's egg. We know we need cells, which comes from the fertilized egg when it divides. We have a place for the embryo to grow in the uterus, and we have the placenta, which allows nutrients and oxygen to reach the human. And finally, we need to know what drugs and medicines are safe and which ones are not safe for the human to grow. We can also study embryonic development to learn about cancer. When human embryos are made, when they're developing, they are highly regulated. All the processes are fine-tuned and the human embryo is organised into distinct structures. Cancer does the opposite. It is characterised by dysregulation and disorder. So why can developmental biology be useful for cancer biology? Embryos and cancers have a lot in common. Embryos come from a single cell, the fertilised egg, and rapidly grow. As it grows, the cells move and interact with each other. Cancer cells do the same thing. The genes in an embryo are turned on and off at the appropriate moments, but reactivation of these genes later on in life causes the cells in the human to move, divide, and interact with each other in abnormal ways. This is cancer. Some genes, which are incredibly important in embryo development, are also regularly seen at abnormal levels in cancer, such as the Wnt genes, the hedgehog genes, and the Notch genes. By studying embryos, we can learn more about how these genes work and hopefully figure out why they are abnormal in cancer. This will allow scientists to develop new therapies for cancer and new ways of detecting it. So now we know some of the basics on how to make a human. But what about part of a human? What if you lose an eye or an arm? They wouldn't grow back. So what can we do to grow back parts of our body? This process is called regeneration. At the moment, humans aren't able to regenerate on a big level, but some animals can. So by studying these animals, we can learn what secrets they have, and maybe in the future, we'll be able to figure out how to grow back parts of our own bodies. Newts are known for their regeneration potential. Newts can regenerate almost every part of their bodies, including the limbs, tail, spine, parts of the eye, the brain, heart, and its jaws. When a newt loses its leg, a lump forms at the wound. This is called a blastema. This will grow to make a new leg. When the newt is forming its new leg, it uses very similar genes and processes as an embryo which is growing a limb. Newts aren't the only animals that can rebuild body parts. Some adult fish can regrow their hearts, some worms can regrow their heads, and deer regrow their antlers. We can learn a lot from animals which are capable of growing new body parts, and combined with studying human development, we may one day unlock the answers for how to regrow parts of our own bodies. Human regeneration is a bit far off, but there are other things we can do to replace damaged or missing parts of the human body. Scientists have been able to grow organs and tissues in the lab. When an embryo is growing, it has some special cells called stem cells. Stem cells are able to develop into lots of different types of cells. For example, when a blood stem cell divides, it can make new blood stem cells, or it can make other types of cells, such as white blood cells, red blood cells, and platelets. In an embryo, these stem cells divide to become all of the parts of the body. In a laboratory, these stem cells can be controlled to form different organs, just like they do in the embryo. As the stem cells divide in the embryo, they can release signals. Neighbouring cells can pick up these signals and use them to figure out what they should divide into. When we have stem cells in the laboratory, we need to mimic these signals that would be present in the embryo. When cells are growing in a petri dish, they all lie flat at the bottom in a single layer. A scaffold can be added to the plate, and this can cause the cells to grow into a 3D structure, and it can act a lot like an organ inside the body. Using this technique, scientists have been able to grow hearts, skin, bone, muscle, and liver. Here is a heart growing in a laboratory, which even beats when electrically stimulated. These lab-grown organs aren't completely perfect yet, but hopefully in the future, they might be able to be transplanted into people who need their organs replaced because of disease or defects. It's also possible to collect stem cells from a person and grow those cells into an organ. This means the organ would be a perfect match for the patient, and they would not need to take drugs to prevent an immune response. These personalised transplants revolutionise the medical field. I really hope you enjoyed this video, and I look forward to answering your questions in the live question and answer session after this video. Thank you.